When I was diagnosed with a degenerative disease, I was 21. Doctors thought I might not survive more than 2 years. Thankfully, I'm still here. A similar prognosis is often faced by people with organ failure, but now, radical forward thinking is offering hope for the future. Replacement body parts to order. A team of scientists in California believe that if you can design them on a computer, you should be able to print them out. Daniel Kraft investigates if there's life in this idea.
One of the most exciting technologies to explode into our everyday world is that of 3D printing. It works on the same principle as your 2D printer at home. But instead of words, the machine can read a three-dimensional design and lay down successive layers of liquid, powder, or sheet material to create almost any shape that can be designed on a computer. In fact, I'm even wearing a 3D printed belt, a single print, and I was scanned by a Kinect and have a version of me, Mini-Me. Now developers are trying to apply extreme 3D printing to a big problem. In the United States, an average of 18 people die each day.
Waiting for organ transplants that don't arrive, there's an organ shortage. I know as a physician that even when organs become available, we need to rush to go and harvest those organs and bring them back for transplantation. What if there was another way to merge science and tissue engineering to make personalized organs available for a patient anytime they needed them and based on their own body?
Sounds like science fiction, but here at Organovo, they're pioneering a way to build human tissue by printing it, cell by cell. Keith Murphy is the co-founder. So Keith, do you envision an era where we're printing our own personalized organs?
Now, we're not quite at the point where we can make full organs, but we can make smaller tissues. We already make blood vessels, for example, and we've already taken stem cells from an individual patient from their fat tissue and built a full blood vessel from that one source of cells.
So how do you build a living blood vessel? The process is remarkably similar to printing my snazzy belt. First, scientists like Vivian design a virtual shape of the tissue they want to print.
I just finished building an example of a tubular structure or a blood vessel. This is a nice interface because it's very easy for the user. For example, as you can see me here clicking through these building blocks that defines whether or not the material is a gel or it's a type of cell, which the printer knows which one to print.
So here it's cell A, B, or C, for example, and those would be different cell types, different sorts of inks,
yes.
Right? Can I build, for example, a different shaped blood vessel, not just a round one?
Absolutely.
I'm gonna try it?
Yeah, go try it.
So even a doctor can design a blood vessel. How many cells would be in each of these circles here?
We're talking in orders of millions of cells for each blood vessel, though.
So this is pretty incredible. This is like a computer-aided design. You might be normally building a square or a picture on a screen, but in this case, we're using the design software to build not just an object, but actually a cellular material, a blood vessel in this case. And this is really, I think, the future of tissue engineering. Three-dimensional, as you think it, you can print it.
This is an ambitious project. From here, the next step is to make bio-ink from living human cells. If they can be printed to form a structure that stays alive, it will be extraordinary. Will it work? "3D printing live human cells has been on the medical agenda for about 10 years. Now, Daniel Kraft is finding out whether one of the leading developers in the field can make it work."
Turning virtual cell structure designs into reality is the challenge. First, you need the raw materials, the bio-ink for the printer. In this case, human liver cells, which remarkably are similar in size to the droplets of ink in an inkjet printer. For this test, we're going to print a mini liver. It will be just 0.5 millimeter deep and 4 millimeters across, but can perform most functions of the real thing. Hi, Vivian. So we've got the printer here. I see 2 different cartridges. What's in the cartridges?
So if you look in this cartridge here, we actually have liver source cells, and in this one here, we have cells that make up a lot of supporting material.
The principle of the bioprinter is similar to how an inkjet printer produces words. Following the computer design, the 3D printer creates sheets of cell-friendly biopaper made from collagen or gelatin to temporarily hold the cells in place. Then, it prints the living cell clusters onto the biopaper, drop by drop. Each drop of this bioink is approximately 250 micrometers wide, fractionally bigger than a strand of human hair. Pinpoint accuracy is essential.
That's one of the great powerful things about 3D printing is that we have the ability to control exactly where the cells go. So just like we said, we can put them exactly where we want to go, and then we're relying on them to do what they know how to do and form tissues.
That's one of the amazing things about this process. Once the cells are in place, nature takes over. Kept alive in incubators, the cell clusters begin to fuse to each other, creating more complex tissue structures.
We can see tissues that we actually printed yesterday that have been incubating overnight.
So from print to incubation to culture in 24 hours. So this is now ready to go for toxicology studies or beyond. The question is, how close a match is the printed tissue? I'm taking a cross-section of human liver to make a comparison. It's a very delicate, thin material. And I'm making 5 microns thick. That's about 1 cell layer. And once it's layered on the slide, I can stain that, put that under the microscope, and see what we've got. So I'm holding a slide of human liver, a thin section that's stained, and you can appreciate the complexity of the liver, which is the major organ in our body which detoxifies our blood. Organovo has printed out human liver cells as well. Human liver, 3-dimensionally printed from stem cells. We'll need a microscope to check the results. How does this compare to a normal human liver? It would look about the same?
Yeah, that's right. So the tissue is highly viable. The cells are happy, they're healthy, they're well organized, and we can do that all exactly in an automated computer-generated process.
The success of 3D bioprinting means that these liver cells can already be used in scientific research to test drugs and study diseases. And because the tissue will have the same DNA as the donor, these tests can be tailored for individual patients. A major breakthrough in medicine. So Keith, where do you see this coming together in the future?
Well, in the far future, the hope for this technology is that we can take a patient's own cells, take them to a central location where we can build them an organ for eventual transplant.
So it's an exciting age for the future of medicine. It's this convergence of 3D printing, tissue engineering, computer-aided design, genomics, all integrating and coming together to enable us to build new organs, new tissues in new, powerful, impactful ways.
That's right.
They've set 2015 as the date to begin tissue graft trials in humans. With bioprinted liver tissue a reality, this thought-provoking field is advancing rapidly. Clearly there is a long way to go before whole organs like the kidney could be printed. I hope that doesn't remain a fantasy.